It's a pleasure to be here, and I will start this talk by doing something you should not do, showing you pictures from my holidays. And the first picture I want to show you, if the slide can go through, let me find this. Yeah, here it's a sea urchin. I hope you can see it. And as you can see on this picture, there's a very nice aster structure, and there's a topological defect at the tip of the sea urchin. You can look other shells on the beach, and this is the one from a famous seashell in France, and there's also a topological defect. And it's not just on seashells. You can look at your head, and there's also a topological defect here. So these defects are actually structural organizers of the body plan of many organisms, in particular plants and animals. And these defects, as we have seen this morning from the talk of Yonit, are also found at a much smaller scale, in particular, in this case, in a monolayer of myoblasts. And there can be different charges, +1/2 and -1/2 in this case. And these defects participate in the velocity and organizing the fluxes of cells in the monolayer. So the question you may ask is are topological defects involved in morphogenesis during development, since they are part of the structure of the animals? And if that's the case, what could be their role during the development? And I refer here to the talk of Yonit this morning that is, I think, one of the best examples we have of the role of topological defects in development. So this talk will be about the work done by Pao in the lab, and he essentially asked two questions. Can we control the localization and the charge of the defects, and can we concentrate the stress so that we can reproduce morphogenetic events in vitro? So what he did was to use a strategy that was used by many groups in the field, and in particular the group of Pascal Silberzan in Paris. And what they did was to concentrate those myoblasts onto circular discs, and you can still see defects in those discs. And if you reduce the size, you reduce the number of defects to 2, the total charge being always +1 because it's a circular defect. Now, if you reduce further, you can even organize those 2 defects into an alignment. And the question that Pao asked was whether, if we confine even more, can we fuse these 2 defects, which are 1/2, into a +1 defect? And these +1 defects should have the form of a ring or of an aster or any type in between, which will be many cases of spirals. So why would you do that? Well, you would do that because these +1 topological defects should concentrate the stress and potentially will lead to multicellular deformation of these monolayers. And that was the goal of the story. So indeed, what Pao observed is that below 200 micron, you can generate single defects, which appear to form these spiraling layers of myoblasts that you see here. Now, these spirals actually transform into asters after a few hours, as you can see here, and that's intriguing. That's a spontaneous transition from one type of defect to another type of defect. So that's caused by increase in density. At least that's, we think, is the driving force. So we could block the increase of density by putting a proliferation inhibitor, which is myosin C, and study the velocity field of spirals, as you can see on the right panel. Now, these spirals, we can also extract the orientation field, and that is shown here. Now, if we let them grow, these spirals will transform into asters, and we can do the same analysis. We can see that in this case, the fluxes are going inward, and the orientational field is essentially the same than the velocity field shown here. Now, these are the averaged velocity and orientational field. The velocity is shown in black lines, and I hope you can see the white vectors that show the orientation field. And as you can see, we can study them with great details, or at least statistical meaningful. Now, what becomes interesting is that those asters actually transform into mounts. They can grow in the center, as you can see on the right panel. And that suggests that the stress field is compressive inside and protrudes the cells from inside outward. Now, one question you may ask is, does the aster configuration is kept during the growth of the mounts? these are some examples of these mounts with a 3D rendering, and you can see that they can really get bigger, and you'll see bigger structures later. Now, we studied in details the alignment, the orientational vector of these layers, and I hope you can see on this part that the bottom layer is actually an aster, whereas if you go to the tip of the mount, you start to see spirals. And this is best represented by the angle between the radius and the orientational vector, which is actually starting at 0 at the bottom of the layer and moves to 60, 70 degrees, which indicates a spiral for the upper part of the mount. So indeed, you have a growth that transforms an aster into a spiral going up. And why this is striking is because those transitions have been predicted in active matter by Carsten Kruse in 2004, and that's the paper they published. And so we went to them and asked whether they could help in understanding what was the stress field here. So with Carles, they started working on the problem, and they included the two main factors in their theory that will lead to formation of spirals and asters. One is directional motility, the fact that cells can go and have a certain persistent movement in one direction. And the other one is anisotropic contractility. These two factors taken separately can lead to formation of spirals, but they don't really lead to compressive stresses. By combining both, they were able to predict various types of asters and spirals with compressive stresses or without compressive stresses, extensile stresses. So you can see an example of extensile stresses in this part of the field. And you can see here that the asters are compressive while the spirals are extensile, but then you can have also compressive asters and spiral at the same time. Now, when we fit the velocity field and the orientational field, as shown here, for various sizes of the spirals with the theory, we can actually predict well the density and the force field, which indeed in this case becomes compressive, as you can see. Can we verify this? So the idea is, how could we measure the local compression in those asters? So Pau came with a very nice idea of building pillars fluorescent, and I hope you can see the 3D images on the right with the cells showing that the pillars are getting compressed by the monolayer of myoblasts. So we can trace this in real time, as you can see from this movie, and we can actually do that for different sizes of pillars, allowing us to measure the stress at different positions on the monolayer. And if we do that, we actually found that the stress increases with time and is more strong in the central part of the layer than on the boundaries. And that is consistent with the theory that predicts this kind of shape for the stress field. Okay, so now we've shown that topological defects in those myoblast layer concentrate states, but can they participate in some physiological relevant mechanisms, and in particular in differentiation or in morphogenesis? So a good thing about this cell line is that it actually spontaneously differentiates into muscle cells from myoblasts to myocysts, as you can see from the myosin marker here. And when we looked where these differentiated cells were in the mounts, they turn out to be in the central part. And you can see the radial distribution on the right panel, and the blue curve is actually the control. And you can see that there's a difference between mounts made like this, where you have a more concentrated distribution of cells, differentiated cells in the center. Now, another good thing that is about with this cell line is that you can isolate sub-cell lines which are not differentiating, as you can see from this. So the left panel shows the cells that are differentiated in yellow, and you can see that you can make layers with non-differentiated cells on the right. So if you use those cells, then you start to see things growing at the bottom of your Petri dish, as you can see on this picture taken by Pau. And these structures become really big, as you can see also on this bright field image taken with the microscope. Now, these are fluorescence images of this, and I call them tornadoes, and Pau called them towers because he's Catalan and they have a tradition of building human towers in Catalonia. So they look very much like this. And these structures can be big. What is interesting is that you can see that it seems to be a topological defect at the top, and the orientational field around here seems to be a vortex. And if you look closer and you do a small analysis, you indeed see alignments of cells around the tower, which suggests that these things are spinning. And indeed, if you look at them with a microscope for hours, you can see that they are indeed spinning with time. And as you will see, if you lose the coating that confines them, then the entire tower drops down, showing that it's really an effect of the confinement. Now, if you analyze this movie, you will indeed see a constant rotation, which suggests that these things are vortices. So what I've shown you today, oh, and I forgot to show you that they can grow big, and if they grow big, then they really get spiral, which also suggests that they are spinning and they are vortices. So I will end up by just summarizing what I've shown you. We start with a spiral, a flat spiral, and through density increase and proliferation, you transform this into an aster. And this transition is reverted by the mount, which grows and reduces the density to form a spiral at the tip of the mount. And this spiral will grow into an aster, it will grow into a vortex and will shape an entire protrusions. So with this, I will stop here and thank people in the lab, in particular, Pau Guillamat, who has done most of the work I've shown today, and of course, Carles Blanche and Carsten Kruse for the theory, which was a very important part of the work. And with this, I will stop and take questions.
Thank you very much, Aurélien, for this very interesting talk. You still had time to talk, so it was, but okay, I have a first question by Vijay. Wait a minute, I will enable your microphone. Yeah, Vijay?
Yeah, hi. Hi, thanks for your very nice talk. So I was wondering, is there a sense of the rotation to the pillars? I mean, is it picked up randomly one way or the other, or there is some chirality in the sense of rotation?
So I cannot say for the pillars or the tower created by the cells, but we have studied that in great details for the spirals. So the initial spiral have, if I remember well, 76% of counterclockwise spirals. And this chirality has been studied for many cell lines in the past. And it seems that indeed there's something, it's never 50-50, it's mostly 60 to 80% of counterclockwise. It seems associated with the chirality of the actin filament as well. At least that's what I heard from scientists from the field.
Thanks.
Okay, I have a written question from Aurélien Roux. Are the vortex tower chiral? Meaning, do they always spiral in the same direction? But
I think it's the same question than the one we just asked. It's a very good question, but we don't have enough statistic at the moment to really tell about the spirals. The vortices, we can say about the spirals, essentially.
Another question from Claire Aquaviva, who question whether what you show with myoblast, is it generally true for other cell types? So
you can generate spirals with other cell types. So for example, Benoît Ladoux has done that with MDCK cells, which are epithelial cells. The fact that we use myoblasts is because they are much stronger in contractility. Of course, they generate much stronger forces. And of course, this is a good system for morphogenesis because you want strong forces for deforming the entire monolayer. By the way, I think you have seen from the talk of Yoni that the alignment of the stress fibers that she sees is probably into the myoblast or the muscular cells of this animal. So that's also consistent with what we know from development.
Okay, Pierre-François is going to ask a question.
Hi, Aurélien. So have you tried to confine the cells on different patterns with different shapes to see whether they form other types of towers, infernal towers?
We have, but for the moment, the only one that grew really well were the circular ones, for the reason I told you. So the initial idea was really to be able to form +1 defects in those monolayer, which is uncommon. Most of the time you get -½ and +½ defects. So it was it was already something to see +1 defect, which concentrate the stress more. If we change the shape, we expect actually not to be able to concentrate the defects into a +1 defect. If we take a square of the same size, you expect to have also -½ defects in the corners, for example, or +½ defects in the corners. So far, what we have tested is the circular ones and the one that worked. We have, I think, tested also the cross, but it didn't give anything positive for the moment.
Okay, we run out of time. I select the last question. So, sorry for the others. We will just use the Slack channel. So from Clara Sidor, what is the consequence of this myoblast behavior in vivo? Do you have any idea? Yeah.
Well, I mean, the idea, and I think our in vitro work is a good connection to UNIT work that we saw this morning. Apparently, it seems that many morphogenetic events seem to be driven by topological defects, whether they're +1 or -½ or +½ and I think the idea is that these defects will organize the stresses that you need for large morphogenetic events. Of course, this is a dream. We are speculating here that it's general, but it has the potential to be a general concept.